We are now beginning Chapter 8 of the Belk et al. Text called Complex Patterns of Inheritance and DNA Fingerprinting. The first section of the book, which includes this portion of the lecture, is Extensions of Mendelian Genetics. We'll talk about incomplete dominance, codominance, and pleiotropy. The next series of lectures will cover sex determination and sex linkage, pedigree analysis, as well as DNA fingerprinting. So the first extension of Mendel, so these are situations in which Mendel's laws did not hold up, are incomplete dominance. So in incomplete dominance, what we know is that neither allele completely dominates the other, and what we see is the expression of both alleles resulting in an average. So if you cross a red flower with a white flower, rather than just getting red or white, as we saw with Mendel, you now get actually a pink flower. So the interesting thing here is if you go ahead and do that cross like we did on the previous slide, the red with the white, and the offspring, the F1s, are going to be pink. If you then cross-pollinate that population, the F1s, what we see in the F2s is that you will get 1/4 as red, half of which will be pink, and 1/4 which will be white. And if you do the Punnett square as we explained when we talked about Mendel's laws, his laws there still hold up to be true. So genotypically it is similar, however phenotypically in terms of what it looks like, it appears to be different. Here's another example of incomplete dominance. So here we're crossing an eggplant and we have this deep purple crossed with a white eggplant, and the F1s are going to be this more lavender colored, and that's the result of them having the big P allele and the little p allele. So take a moment and try to determine what the F2 generation would look like. What would they look like in terms of their genotype, and what will they appear to look like in terms of their phenotype? So put the video on hold for a second and try and figure that out yourself doing a Punnett square. So hopefully your results look something like this. 1/4 of the offspring, if you crossed the F1 generation, would be that deep purple. Half would be, or 2/4 would be that lavender, softer violet color, and 1/4 would be white. So this is incomplete dominance. Here's another example that we see in nature. If you cross this white horse with the brown horse, you actually get this really beautiful kind of more palomino colored horse. And this is an example, again, of incomplete dominance. So here's the next extension of Mendel. So we've talked about incomplete dominance, and now we're going to briefly talk about codominance. And this is a situation in which both alleles are equally and completely expressed. This is what we see for blood typing. And there's also, this holds true for many different types of animal coat colors, and that's how we get oftentimes spotted animals. So here, what we've done is crossed a red flower with a white flower, and the offspring are this red and white flower. The offspring do not look like either parent. The offspring here is a red and white flower. It doesn't appear to be all red or white, which is what the parents were. So that's the situation which we have for blood groups and blood typing. So blood typing is somewhat complicated. There's actually 3 alleles that play a role in the determination of your blood type. If you have allele A or allele B, both of these can be codominant to each other, whereas little I is recessive to both of the other alleles. So any individual is just going to have 2 of these alleles. So for instance, if you are big A, you are going to be type A. If you are big A or with little I, your blood type will be A. This is in contrast to, say, A with B, in which case your blood type is going to be type AB. Or if you have little I, then your blood type is type O. What do these alleles for the different types of blood actually encode for? Well, they are simply a sugar that gets attached onto a protein that's in the plasma membrane of red blood cells. So if your genotype has the A allele, then you are putting on the proteins in your plasma membrane a specific type of sugar attached to that protein. Here it's depicted in red. If you have the B allele, you are placing onto that protein a different type of sugar. If you are type AB, then you're presenting on the proteins, on the red blood cells, both types of sugars. If you have the little I allele, then you actually have a mutation in this gene and you do not place any sugars on the attached proteins. So if you cross AA individual with a BB individual, you will get AB, and we know that these are co-dominant. Those sugars that get presented, you're going to be presenting both types of sugars on the proteins on the red blood cells. If you cross AA with OO, it will be an individual that is A blood type. They're going to be presenting just one type of sugar. If they're BB crossed with O, they will be considered B blood type and just presenting that second type of sugar. We say that A and B are co-dominant. A is dominant over O and B is dominant over O. Therefore, O is recessive to both A and B. So when we're talking about dominance, you can either have complete dominance, which would be the case that we saw for Mendelian traits. You can have incomplete dominance or co-dominance. If it's complete dominance, what we'll see is when you cross one parent with another parent, if all the offspring for that particular trait look like one parent, then that is an example of complete dominance. If you cross, in the case of incomplete dominance, one parent with another parent and the offspring actually are a hybrid or a mix of what either parent were but don't actually look identical to either parent, we call that incomplete dominance. If we cross, in the case of codominance, one parent with another parent and you see both of the phenotypes present, we call that codominant. The last topic to discuss is pleiotropy, and this is when a single gene has multiple effects on the individual's phenotype. So for instance, with regards to hemophilia, which is basically an inability to clot your blood due to the absence of a particular clotting factor, we know that this gene is on the X chromosome, but it's not only involved in blood clotting, and phenotypically we see this exemplified as general weakness, inability to clot blood, as well as easily bruising. So even though it's just regulating one gene, phenotypically you see it presented through lots of different mechanisms. So in summary, we've talked about incomplete dominance, which is basically a blending of your traits, codominance, in which both traits are equally expressed, and pleiotropy, which is just a mutation of a single gene, but it affects phenotypically many different symptoms.